Hello guys, welcome to my YouTube channel Ambitious Dr. Ashwini, and today we will see the next protozoa, that is Plasmodium species. So Plasmodium comes under Sporozoa. You can see Plasmodium malariae comes under Sporozoa. So in this the contents are: describe the life cycle of Plasmodium species, describe the pathogenesis of Plasmodium species, describe clinical features of malaria, and explain the laboratory diagnosis of malaria. So we will see it by one. First one, introduction. Geographical distribution. It occurs in all countries in the tropical and subtropical regions. Plasmodium ovale is not reported in India, whereas Plasmodium vivax, Plasmodium malariae, and Plasmodium falciparum exist in India. Habitat. It is found in parenchymal cells of the liver, erythrocytes, and other organs. Agents. Human infection is mainly caused by five species: Plasmodium vivax, Plasmodium falciparum, Plasmodium malariae, Plasmodium ovale, and Plasmodium knowlesi. So first, Plasmodium vivax. It causes benign tertium malaria and the fever recurs every third day. Plasmodium falciparum causes malignant tertium malaria. It is the severe malaria and fever recurs every third day. Plasmodium malariae. It causes benign Corton malaria. The fever recurs every fourth day. Plasmodium ovale. It causes oval tertian malaria. The fever recurs every third day. Plasmodium knowlesi. It causes quotidian or simian malaria. The fever recurs every day. It is a parasite of monkeys but can also infect humans. In short, third day fever occurs in Plasmodium vivax, Plasmodium falciparum, and Plasmodium ovale. Fourth day fever in Plasmodium malariae and daily fever occurs in Plasmodium knowlesi. Now we will see the Plasmodium life cycle and malaria. So the life cycle of Plasmodium species. So Plasmodium completes its life cycle in two hosts. So the hosts are definitive host, that is female Anopheles mosquito. The sexual cycle or sporogony takes place. Second one, intermediate host, man. Man is the intermediate host. The asexual cycle or schizogony takes place. Transmission to man and infective form. When an infected female Anopheles mosquito bites a person, it injects the infective form called sporozoids from its salivary glands into the blood. Rarely, malaria can also spread by blood transfusion and from mother to baby, that is transplacental. In such cases, the infective form is tropozoids or myriazoids. Human cycle. In humans, the asexual cycle takes place through three stages: pre-erythrocytic stage, erythrocytic schizogony, and gametogony. So, firstly, we will see pre-erythrocytic schizogony, which occurs in liver. Sporozoids enter the liver within 30 minutes after a mosquito bite. They attach to hepatocytes, which are the liver cells, using circumsporozoid proteins. Inside, they changes into tropozoids to schizons, which contain many myriazoids. On liver cell rupture, myriazoids are released into the blood, starting RBC cycle. In Plasmodium vivax and Plasmodium ovale, some sporozoids remain dormant, means inactive or in sleeping state, in the liver as hypnozoids, which causing relapse years later. Relapse versus recrudescence. So relapse means reactivation and recrudescence is return. So relapse is due to hypnozoids, which occurs due to Plasmodium vivax and Plasmodium ovale, and recrudescence due to survival of a few parasites into the blood, which is due to Plasmodium falciparum and Plasmodium malariae. Second stage is erythrocytic schizogony, which occurs in the RBCs. Here, myriazoids enter RBCs by binding to glycophorin receptors. Inside RBC, the early stage ring forms in tropozoites. Later, schizons produces new myriazoids. RBC rupture, releasing myriazoids, infects new RBCs, causing repeated cycle of fever. Plasmodium feeds on hemoglobin, releases waste, that is hematine plus iron porphyrin, which is malaria pigment. Time taken for one RBC cycle. Plasmodium vivax, Plasmodium falciparum, and Plasmodium ovale takes 48 hours to complete its RBC cycle, and Plasmodium malariae takes 72 hours. Plasmodium falciparum later stages hidden in capillaries of brain or internal organs. Only rings forms seen in peripheral blood smear. And the last stage is gametogony. Sexual forms in humans. Some myriazoids in RBCs develop into gametocytes, which is the sexual forms. Two types: microgametocytes, which is in male, and macrogametocytes in females. Shape is round in all species, but in Plasmodium falciparum, the shape is crescent or banana shaped. These gametocytes are not for humans but are the infective stage of mosquitoes. So, the sporozoids, you can see, sporozoids enter the liver within 30 minutes, then in hepatocytes, tropozoites, and schizons, it ruptures and releases periozoites. It goes to in erythrocytes, that is RBCs. Here, tropozoites, the ring like formation happens here, then schizons, then myriazoids releases, and it again infect the erythrocytes. So, this is the fever stage, that is RBC stage. Then from tropozoites to gametozoids, here production of gametocytes happen, that is microgametocyte, that is male, and macrogametocyte, that is female. Then it is ingested by the mosquito. So this is the life cycle happens in human. Now we will see the life cycle which happen in malaria, mosquito. So in short, liver stage, that is pre-erythrocyte stage, sporozoids release, tropozoids, schizons, and myriazoids. In RBC stage, that is erythrocytic. Myriazoid, the ring form happens in tropozoids, then schizons, rupture, and fever cycle occurs. Sexual stage, that is gametogony. Myriazoids, gametocytes, which infects mosquito. Now we will see the mosquito cycle of Plasmodium, that is sexual cycle or sporogony. When a female Anopheles mosquito bites an infected person, it takes in both asexual form and gametocytes. The asexual forms is digested and gametocytes continues development. Now we will see the stages. Gametocytes in the mosquito gut. Male gametocyte undergoes exflagellation and produces 8 male gametes. Female gametocyte directly transforms into one female gamete. Here fertilization occurs, male gamete unite with female gamete and form the zygote. Zygote elongates motile and forms the motile ookinete. Oocyst formation. Ookinete penetrates the mosquito's stomach wall and develops into oocysts. Sporogony. Inside the oocysts, many sporozoids are produced. Sporozoids migration. Mature oocyst ruptures, sporozoids are released. Then sporozoids migrate to mosquito salivary glands and ready to infect humans in next bite. The important points are: time taken for this development inside mosquito is extrinsic incubation period, which is 1 to 4 weeks and varies with species. In short, gametocytes forms the gametes unite to form the zygote, fertilization happens and zygote is formed, then ookinete, then oocyst, sporozoids released into the salivary gland. So from here The microgamete and macrogamete are formed, which are uniting to form the zygote. Here, fertilization occurs. The ookinete elongated shape happen here, which penetrates the stomach wall of the mosquito, and the oocysts are formed. In this oocyst, many sporozoids are there. These are released into the salivary duct of the mosquito, and the life cycle goes on. Now we will see the pathogenesis of Plasmodium species. Malaria is caused by the protozoa parasites of the genus Plasmodium, transmitted to humans through the bite of infected female Anopheles mosquitoes. The pathogenesis involves sporozoid invasion. Upon mosquito bite, sporozoids enter the bloodstream and travel to the liver. Liver stage: sporozoids infect liver cells, mature, and reproductive asexually to form myriazoids. Erythrocytic stage: myriazoids are released into the bloodstream, infect red blood cells, and undergo asexual reproduction, leading to RBC rupture and the release of more myriazoids. This cycle causes the clinical symptoms of malaria: gametocyte formation. Some myriazoids develop into sexual forms, that is, gametocytes, which are ingested by mosquitoes during a blood meal, continuing the transmission cycle. Now we will see the clinical features of malaria. The incubation period of Plasmodium falciparum is 12 days, Plasmodium vivax is 14 days, Plasmodium ovale is 17 days, and Plasmodium malariae is 28 days. Benign malaria caused by Plasmodium vivax, Plasmodium malariae, Plasmodium ovale, and sometimes mild falciparum. Now we will see the triads of features. First one, febrile paroxysm, which is intermittent fever. Fever occurs when myriazoids are released after each RBC cycle. Fever pattern: every 48 hours, that is third day, Plasmodium vivax, Plasmodium falciparum, and Plasmodium ovale. Every 72 hours, fourth day, Plasmodium malariae. Each paroxysm has three stages: cold stage, that is shivering and chills happens; hot stage, high grade fever; sweating stage, profuse sweating with fall of temperature. Second clinical feature is anemia due to the destruction or lysis of RBCs. Falciparum severe anemia infects all ages of RBCs. Plasmodium vivax and ovale infect young RBCs or reticulocytes, which are immature RBCs. Plasmodium malariae infects old RBCs. And third one is splenomegaly. Spleen enlarges due to overactive macrophages, immune cells removing both infected and damaged RBCs. So, in short, the clinical features: benign malaria triad, fever 48 or 72 hours cycle, anemia, and splenomegaly. So these are the three stages of malaria: cold stage, hot stage, and sweat stage. Now we will see the laboratory diagnosis of malaria. So the diagnostic tests of malaria can be divided into microscopic test and non-microscopic test. So firstly we will see microscopic tests. Peripheral blood smear is the gold standard test. Here thick smear, which is more sensitive; thin smear, speciation, can be done based on the following features, that is Plasmodium vivax, amoeboid ring form and schizont; Plasmodium falciparum, ring forms, multiple ring forms, acole form, headphone shaped ring forms, banana shaped gametocytes; Plasmodium malariae, band forms; Plasmodium ovale, enlarged fimbriated oval RBCs with ring forms. Quantitative Buffy coat examination: parasites, RBCs appears as brilliant green dots. Next is non-microscopic test. Antigen detection test, RDTs or ICTs, detect pan-malarial AG, LDH, aldolase. Falciparum specific AG, HRP second. Culture RPMI 1640 medium, molecular diagnosis PCR targeting 18S rRNA. So here microscopic diagnosis, antigen detection and molecular diagnosis is done. Thank you. Please like, share and subscribe to my YouTube channel and don't forget to press the bell icon for latest updates.